All right, I think we'll get started. And so good evening, nurse colleagues. And I think we've got some nursing students also listening. So welcome to, I think is going to be a pretty dynamic webinar on the topic of messenger RNA, the path from basic science to pandemic vaccine. My name is Gina Denick-Champion. I'm the Executive Director of the Wisconsin Nurses Association. And on behalf of our WNA Board of Directors, I want to share how pleased we are that you have joined us for this obviously very relevant and informational presentation. Next slide, Bree, thank you. So I just want to, just a couple of things. So we will be awarding contact hours for you during this live webinar. We would very much like for you to complete the evaluation. That's how you will get your contact hours or what you can use to show that you attended. But if you can please complete the evaluation by Friday, November 12th. The link is on this slide, but at the end of this presentation, you will be getting another email with the instructions for how to join or get to the link for this evaluation. And we would very much like your feedback. Next slide. Thank you, Bree. So, and in terms of questions and answers, we will have some time for questions. Some folks have already submitted some, and Dr. Balagia has already reviewed those. But if you have questions, please use the chat box. And I will be reading the questions that come forward. We will try and address as many questions as possible. And what we will do is take the rest of the questions in the chat box, and we will put those into our networking platform called Wisconsin Nurses Connect, where we can continue the conversation after that. So that's available to all members, and we'll include that link in the near future. So with that, it is my pleasure to, next slide. It is my pleasure to introduce Dr. Edward Balagia. He is, we're really lucky that we have him tonight. He's an infectious disease epidemiologist and currently serves as the director of the Center for Clinical Epidemiology and Population Health at the Marshfield Clinic Research Institute. He has led CDC-funded studies of vaccine safety and vaccine effectiveness for over 15 years. He is a former member of the CDC Advisory Committee on Immunization Practices and currently serves on the CDC COVID-19 work group. Dr. Balagia graduated, I'm sorry, I was slaughtering that, graduated from Michigan State University College of Human Medicine. He completed his residency in internal medicine at UW Hospital and Clinics and has served as an epidemic intelligence service at the CDC. And like I said, we're very fortunate to have this caliber of expertise joining us tonight, and also that that we have him here in Wisconsin. So I'll turn that over to you, Doctor.
Okay, thank you very much. I'm going to share my screen here quickly, and hopefully this will work. All right, can you see my screen?
Yep.
Yes.
Yes.
Yep.
Okay, great. Okay, well, thank you for that very nice introduction and for inviting me to speak. I very much appreciate that, and I think it's especially important to talk to people who are in healthcare who have questions and concerns and try and address those so that people have accurate and up-to-date information. And my goal here is to finish this so we have time to have quite a bit of Q&A and discussion at the end. And also, I guess I just would say that it's been a real privilege for me to sort of see vaccine development and research from sort of two different angles. One is as a researcher where we've been doing vaccine safety and effectiveness research, largely with CDC funding for the past couple of decades. And then also from 2014 to 18, I was a member of the CDC ACIP, which sets national policy and makes recommendations to CDC regarding vaccine policy recommendations. And so I got to see a little bit of how the sausage is made and be part of that, and sort of the difficult decisions that have to be made sometimes with incomplete data. So it's given me, I think, some useful insights, and I'll try and share as much of that as I can with you over the next 40 minutes or so. So I'll go to the next slide. Let's see. There we go. So what I want to talk about today is a quick overview on coronavirus disease in humans and how the immune system responds to coronaviruses. And then spend a fair amount of time on mRNA vaccine development and how that came about. And then just kind of give you a high-level overview of what is an emergency use authorization and how is that different from licensure, and sort of how are we moving through this process. And then a high-level overview with some current information on vaccine effectiveness, especially in the Delta variant time period, and vaccine safety monitoring and how we do that and what we're learning. And then just kind of close with just a couple of slides on misinformation and disinformation, which is rampant, and some suggestions as to how to navigate that. Okay, so coronaviruses have been known to cause disease in humans for a long time, and there are 4 different subtypes that are known to cause mild seasonal respiratory illness, basically common colds, mostly in the winter. But there are many other types of coronaviruses that circulate in mammals, especially in bats. The first serious human threat from coronaviruses was back in 2003, and I remember that quite well at the time. That was the Sars outbreak that affected 28 countries. Ultimately, there were over 8,000 cases and over 700 people died. And the case fatality ratio was pretty high. It was around 15%. We dodged a bullet with that one because with a very aggressive public health response, it was eradicated, and everyone breathed a sigh of relief. Until 2012, when another coronavirus popped up in the Arabian Peninsula, and that was the Middle Eastern Respiratory Syndrome virus, or MERS. And that has an even higher case fatality ratio compared to SARS, and it is ongoing. But fortunately, it does not spread from person to person. It is largely a zoonotic disease. It is acquired from camelids. And so it remains a scourge and a problem there, but it has not been a pandemic threat. And then, of course, we all know what happened in December of 2019 with the outbreak of novel coronavirus in Wuhan, leading to the pandemic. Well, I want to point out a couple of things here. First, in terms of what we've learned. We learned quite a bit about coronaviruses, SARS coronaviruses in 2003, and the current SARS-CoV-2 is very closely related to the original SARS with about 76% amino acid sequence match. And in fact, the cartoon on the left here shows the structure of the spike protein on the surface of the original SARS virus, published back in 2006, and it attaches to the same receptor in human cells. And on the right side is a graphic of the SARS-CoV-2 spike protein that was published in early 2020. And so we understood quite a bit about how this virus works, how it gets into cells, and what type of immune response it generates even before the pandemic occurred. And that's important when we get to talk more about vaccine development. So there are a number of different ways that sort of vaccines have been historically developed, going all the way back to Jenner and cowpox. And on the left-hand side of this slide, you see what's been sort of the classical vaccine production that accounts for a lot of vaccines that are still currently used today. We have inactivated vaccines, things like rabies, flu, polio, Hep A, where you essentially take the pathogen and you kill it, inactivate it, and you break it up and purify the parts you want and then inject it. So it's a pretty basic process. And then on the bottom side, on the left, we have live attenuated vaccines, which are represented by vaccines like smallpox, the MMR vaccine, varicella, and yellow fever, where you take an actual virus, but you attenuate it, you weaken it, so it's not able to cause illness, but it does generate an immune response. And both of those approaches work generally, and they've been pretty successful, but now we're in the era of structure-based vaccine design on the right, where in fact we can sort of target the vaccine to produce a protein that generates the immune response that we want to get. And there are different ways that we can do that. We can use adjuvants, which sort of boost the immune response. We can use recombinant technology. We can generate nanoparticles or virus-like particles. And in particular, we can use new strategies like mRNA and viral vectors. So that's what I'm going to focus on, especially mRNA. So just to give you a little background on mRNA. mRNA is something that all cells use to produce proteins that keep cells functioning. Natural mRNA was first discovered in the 1960s, and it essentially provides the assembly instructions for cells to manufacture proteins based on the instructions coming from the DNA. In the 1980s, though, was the first time that an experiment actually demonstrated that if you take mRNA and you mix it with sort of a soup of fat, you can actually get cells to produce the protein that the mRNA is coding for. So that sort of raised some eyebrows and interest in whether this could be used as a therapeutic to basically have an mRNA produce drugs in the body or vaccines. And in the case of the current vaccines, mRNA is delivered in a lipid nanoparticle, and then the cells translate that mRNA sequence into the protein, which is the spike protein that triggers the immune response. And you can see this in the cartoon on the right, which is from a Mrna presentation at an FDA meeting. And again, in the case of the current mRNA vaccines, the mRNA is rapidly degraded within a day or two and does not replicate, unlike viral infections. So that sort of is another point I really want to emphasize here, that people who have thought that this mRNA process is very foreign, it's very weird, it's not natural. But the reality is every time you get infected with a flu virus or RSV or some other respiratory virus, what we're describing here is exactly what's happening when that virus is going into your body and it's getting into your cells, and the virus gets into your cells and it uses the mRNA in your cells to make copies of itself. And those copies then, in this case, we're not giving an entire virus, we're just giving the sequence for one single protein, so it's impossible to get an infection. But the basic process of sort of using mRNA to produce the protein and then that's expressed on the cell is the same as when you get a viral infection as with this, except in the case of these vaccines, you don't actually get any infection at all. Okay, there are a number of milestones in the history of mRNA vaccine development. As I mentioned, they discovered that you could actually get mRNA to manufacture proteins if you delivered it to cells. And another breakthrough was that we learned how to actually synthesize it outside of the cell. Before that time, you had to basically harvest mRNA in order to use it rather than synthesize it. So we learned how to synthesize it. We learned how to deliver it into human cells using liposomes. And then in the 90s, mRNA began to be tested as a therapeutic treatment in rats. And that was the first also attempt to actually test an influenza vaccine in mice using mRNA. Not a lot of progress was made, though, in the 90s, and there were some big barriers to making this work in humans that I'll talk about in just a minute. In the 2000s, there was a major discovery that that you could modify the RNA to reduce inflammation and improve stability, because a huge barrier to using this was the fact that the mRNA is an unstable molecule, so it did not survive well if you injected it. And it also triggered inflammation in people. And so it took some time to figure that out. Another major development that occurred in the 2000s was a scalable method to manufacture lipid nanoparticles, which were essential to be able to get the mRNA into the body and keep it from being broken down and triggering an immune response. So then in the past decade, the first mRNA vaccines in lipid nanoparticles were tested in mice. And then actually, again, this is all before COVID-19. There were human trials that were initiated for mRNA vaccines in lipid nanoparticles, and these include trials for vaccines against influenza, rabies, Zika, and CMV. So the saga of mRNA vaccines is filled with many people contributing, and I'm sure there are hundreds, if not thousands, of people who have made very important contributions to this. And I just want to highlight a couple of them in particular, because I think the story is fascinating, and I do think their contribution is such that they may well end up getting a Nobel Prize for their work, even though many others made major contributions to what ultimately led to these mRNA vaccines. These individuals are Catalin Carrico, who's currently at BioNTech in Germany, and Drew Weissman, who's at the University of Pennsylvania. It's a fascinating story. Dr. Carrico actually grew up in Hungary in a small village. Apparently they didn't even have running water in the village, or in their house, and but she was fascinated by science. She got her degree in Hungary and then came and did a postdoc in the United States, and wound up with a faculty position at the University of Pennsylvania. And all along this way, she was very interested in the potential to use mRNA as a therapeutic or as a vaccine, but was struggling with the problems I was describing earlier in terms of inflammation, and frankly, she couldn't get much funding either. This was a backwater. It was not considered a very promising area of research. She got turned down on many grant applications and was denied promotion at the University of Pennsylvania. So not a very auspicious sort of start, and yet she really persisted in this area, even though she was really very discouraged from doing this. Well, sometime in the late 90s, she met Drew Weissman, shown here in the photo on the top right, and actually met at the copy machine, which back then was how you stored articles. And he was working in a different area. He was an immunologist with a particular interest in HIV vaccine development and hadn't really thought much about mRNA. And the two of them started talking, and with his knowledge of immunology and her knowledge of mRNA, they started developing some interesting experiments and made some amazing progress. And they published this work, and some of it you're seeing here on the right-hand side of the slide. First major one was in 2005, where they basically learned that you could make a minor modification to one of the components of the mRNA, and they basically took uridine and modified it to something called pseudouridine. It's a naturally occurring modification that occurs in the human body, but it's just not in messenger RNA in the human body. So it's not a foreign molecule at all. But by making that modification in the mRNA, it reduced the inflammation and it improved the stability of the mRNA. So they could deliver it, and it would not trigger an inflammatory response, and it would get into the cells. That was a major development in 2005. And there was a very nice sort of bio in the Washington Post on October 1st of Catalin Carrico, and the author sort of describes this as how they made what they thought was this major breakthrough based on a minor tweak to the genetic code of the mRNA that really changed everything and made it much more feasible. And they published this, and then there was silence, like nobody cared. And it didn't really lead to any more grant funding. And eventually she left, and she left the University of Pennsylvania. She didn't get promoted, and she took a job in around 2013 with BioNTech, where they were very interested in mRNA development. And on the bottom of this slide, you can see some other studies that they published. One in 2008 showing that there is increased stability of using the pseudouridine instead of uridine in the mRNA. And then in 2017, they published in Nature actually a proof of concept, then going from just the laboratory to actually showing that in mice you can actually protect them against Zika virus infection with this modified mRNA vaccine. Okay, so I want to segue and talk a little about the accelerated COVID-19 vaccine development, because again, one of the concerns that we hear over and over again is they rushed this through, it went too fast. How do we know it's safe? They cut corners. And I want to try and explain to you why that's not the case. First of all, if you look at the bottom of the slide, it shows the process that a company has to go through to get an investigational new drug licensed. First, they have to do animal studies, preclinical studies, and then they apply for an IND, and they go through phase 1, phase 2, phase 3 studies with increasing numbers of people, starting first just with a safety outcome and then sort of moving more into immune response and ultimately to efficacy against preventing infection. And then they apply for FDA review and approval, and then if it's licensed, they go into phase 4. And throughout the whole process, the FDA prioritizes safety, efficacy, and manufacturing consistency. Now, this process normally takes a decade or more, and it costs hundreds of millions of dollars. And so companies, because they are basically putting their own money at risk, they move through this in a very methodical way, and they don't go from phase 1 to phase 2 until they've fully vetted the fact that it's worthwhile to go from phase 1 to phase 2. And that makes the process take a long time. So what do we do to sort of accelerate this in the face of a public health emergency? Well, one thing was we had the advantage of a knowledge base from prior SARS and MERS vaccine research that was done back in the 2000s after the original SARS and then after MERS in 2012. So we already knew a fair amount. We knew about the spike protein. We knew about the type of immune response it elicited. So we had a head start in terms of developing a vaccine. Another key factor was the public-private partnerships and the industry collaborations that went on. And I'm sure you recall Operation Warp Speed, which was a US government funding that basically allowed companies like Mrna to get hundreds of millions of dollars to really accelerate their development and to work with NIH to do that. And there were a number of companies that got federal funding. Another key component within the industry was smaller companies that really had the cutting edge expertise on mRNA, like BioNTech, partnered with much larger companies like Pfizer that had both the bandwidth, the resources, and the expertise to do large-scale vaccine manufacturing and R&D. And then another factor was basically, again, to compress the timeline without cutting corners. Instead of doing phase 1 and then going to phase 2 and then going to phase 3, they compressed the trial phases, and they wrote protocols that would be combined phase 1, phase 2, combined phase 2, phase 3, all in one protocol, and to accelerate the process. And then lastly, and I think very importantly, the US government basically took the financial risk away from the manufacturers by agreeing to buy doses and allow manufacturing to be scaled up even before the human trials were completed, so that once the trials were done and the data were assessed, if the vaccines were found to be safe and effective and they were authorized, they could be distributed almost immediately rather than waiting months to begin to manufacture them after they were approved. So all of these things allowed these vaccines to develop and rolled out in record time. And the time from the first public reporting of the sequence of the virus from China in early January until actually having a prototype vaccine at Mrna was only 2 or 3 weeks. And it was less than 100 days before the first vaccines were actually being injected into the arms of volunteers in clinical trials. And that is astounding, and I think that's a real credit to everybody that was working on this. Okay, so now I want to just talk a little bit about the regulatory issues. As you know, these vaccines were originally used under emergency use authorization. This is a clause that allows the FDA to allow use in a declared emergency where the benefits are deemed to outweigh the risks. Now, even though this is an emergency use authorization, the FDA still requires the manufacturer to submit a full application with all of the data on efficacy and safety. The FDA will independently reanalyze the data. They do not just trust the analysis done by the manufacturer. For the original EUA, they required a safety follow-up for 2 months, and they required vaccine efficacy of 50% or more. And this is standard. The FDA for all vaccines and the CDC have independent advisory panels that review their data and their decisions and make recommendations. So on the right-hand side of this is the Biologics license application, which is the more standard process. And in that case, they would generally have longer follow-up on the clinical trial data, typically for at least 6 months or longer. And there would be additional review of manufacturing practices and facilities. They also reanalyze the trial data that's submitted by the manufacturer, and there's also independent review by VRBPAC and ACIP. Okay, so here's kind of the current situation. We have one vaccine that is fully licensed. That's the Pfizer-BioNTech vaccine, and that's 2 doses, 3 weeks apart, and it's licensed for age 16 and up. It was licensed in August, so it has met the full bar for licensure. But we still have a number of products in situations that are where we're still operating under EUA. Pfizer itself still has EUA authorization for age 12 to 15, as well as giving a 3rd dose to people with moderate to severe immune compromise, because it's clear that those folks do not respond nearly as well.
Yes.
Okay, I just heard a voice tell me recording was stopped. I just wanted to make sure my voice didn't stop, so thanks. And then Mrna is still under EUA. They have applied for licensure, but the FDA has not yet acted on that. And then the Janssen vaccine, which is the adenovirus vector vaccine, is also still under EUA. So, and then just briefly mention the booster authorizations. Both Pfizer and Mrna are authorized for a single booster dose at least 6 months after completion of the primary series. For people 65 and up, people 18 to 64 at high risk of severe COVID-19, and people 18 to 64 with frequent institutional or occupational exposure to SARS-CoV-2. And then the Janssen vaccine was also authorized as a single booster dose at least 2 months after completing the primary series for age 18 and up. And importantly now, because of an NIH clinical trial, which the results have been released and reviewed by FDA and CDC, showing that, in fact, if you mix and match vaccines, you get at least as good of a response, and in some cases, you even get a better response than if you give the same vaccine as a booster. That CDC allows that mix and match booster, so you do not necessarily need to get the same thing that you got with your primary series when you're getting a booster, and that really greatly simplifies the distribution of boosters. You don't have to worry about that anymore. So that was really good news. Okay, let's just talk about vaccine effectiveness for a little bit here. And I want to highlight a few things about the vaccine-induced immune response. So we're all on the same page on this. One of the most important components of the immune response is generating neutralizing antibodies. And neutralizing antibodies are ones that specifically inactivate the virus. And we know that higher levels of neutralizing antibodies are strongly associated with higher levels of protection. But another component is something called the cellular immune response, where you develop a memory B cell response. So if you get re-exposed in the future, you can very rapidly ramp up your immune system to respond to the reinfection. And your T cells play a key component in reducing the severity of illness, and so your cellular immune response is important on reducing severity. And another key component is that we know that older adults have immune systems that do not function as well, especially their cellular immune response does not function as well as younger people. And that is one of the reasons why they probably get more severe illness. And so overcoming immunosenescence is a key for vaccines. And then the issue of sterilizing immunity, that is, it's clear at this point that the vaccination reduces your risk of infection, it reduces your risk of serious illness, but it does not generally cause sterilizing immunity, because we know now that vaccinated people can, if they become infected, they can shed the virus and transmit the infection. Although I want to emphasize that people who are vaccinated have a much lower risk of becoming infected in the first place compared to unvaccinated people. So I'm just going to show you a little bit of studies of vaccine effectiveness which support the need for a booster, at least in some populations. This is a CDC slide showing several studies looking at vaccine effectiveness against infection over time in adults from February up through July. And then you can show that you see the Delta period in June and July. And you can see that multiple studies do see some modest decline in overall vaccine effectiveness coinciding with the period when the Delta variant was rising. This is another study from the CDC Vision Network that shows vaccine effectiveness against hospitalization by time period and by age group, and separately for Pfizer-BioNTech on the top and Moderna on the bottom. Let's just look at the top here first. This is the Pfizer data. And if you start on the left, that's people 18 to 29, and you see 82, 85% protection, so about the same from January to May versus June to August. And then if you look at the middle of this figure, it's age 30 to 49, it's about the same, around 80 to 85%. And then moving to the right, then we see 84 to 93% for people 50 to 64. And then on the right, far right, these are people 65 and up. On the top, that's Pfizer. It looks like vaccine effectiveness has declined somewhat against hospitalization from January to May, going to June to August. And on the bottom, you see the data for Moderna. You don't really see as much of a decline for Moderna. Moderna is holding up a bit better. And overall, across the ages, we see pretty high and consistent levels of protection with Moderna. I want to also point out this is the Heroes Recover study, and this includes healthcare personnel and first responders. And so given the audience tonight, I thought this would be of interest. And this shows overall VE against infection. And you can see that the adjusted VE against infection on the top for the overall cohort was 80%. But if you look at the time periods after dose 2, it's 85% for people who are less than 120 days, 81% for 120 to 149, and then it goes down to 73% for people over 150. And then on the bottom side of this, if you look at pre-Delta versus Delta, it looks like it's gone down during the Delta period. the horizontal lines represent the confidence intervals. So from that we can say these are not really super clear data because there's a lot of overlap. Statistically, the picture is a little bit fuzzy here. But I think the point is that there are a number of studies that are consistently suggesting that there's some modest reduction in vaccine effectiveness, including hospitalization in older adults. Okay, and so I also want to bring in the Israel experience because Israel rolled out their vaccine to their entire population faster than most other places. And they're exclusively using the Pfizer-BioNTech vaccine. And they noticed a substantial spike in the incidence of cases and hospitalizations early in the summer. And so they basically rolled out booster doses in August, starting with people over 60 and then progressively rolling it out to people over 50, and then ultimately to everybody over age 12 was eligible to get a booster dose. And they've been tracking their occurrence of both cases and hospitalizations very carefully. And they have a very nice system where they can link data for their whole population. So they can do studies more quickly than what we can do in the US on this. And they're seeing a 12-fold reduction in the rate of infection after the booster dose in people over 60 compared to people not boosted. They're not seeing any safety concerns. In particular, they are not seeing any evidence of myocarditis occurring at any higher rate in people getting boosted. And the figure on the right here just shows the absolute rate of infection in people not boosted in yellow, and then people boosted in brown, I think, whatever that color is. And so you can see the rate is much, much higher in the people not boosted. And then this specifically looks at severe disease rates in people over 60, and they see a 5-fold reduction in the risk of death in people after being boosted versus a comparison window immediately after the booster when there's no biological basis that there would be any booster effect at all. So as an epidemiologist, I would say this is a pretty strong study design that's not going to be subject to too much bias, and they're seeing a substantial benefit to the booster in people over 60. So this also was taken into consideration by the FDA and by the CDC advisory panel. And also, I just want to mention Wisconsin, and specifically, Wisconsin, I think, has some great stuff on their website. And this is a graphic that's on the DHS website showing basically a visual representation of the rate of developing COVID-19 among people in Wisconsin who are fully vaccinated and people not fully vaccinated in the month of September. So blue shows of 100,000 fully vaccinated people, it shows the number of cases that were seen in Wisconsin. Again, this is a rate; these are not absolute numbers. And the darker blue shows 14 hospitalizations per 100,000 people, and basically 1 death per 100,000 vaccinated people. And then, sort of in orange on the right, or red, among people not fully vaccinated, the rate of developing COVID was 2,300 cases per 100,000 people, 122 hospitalizations per 100,000 people, and 24 deaths per 100,000 people. So more than 20 times as many deaths in the unvaccinated, not fully vaccinated versus fully vaccinated. So the difference is real and very profound. And I just threw the slide in just based on reading some of the questions that came through today. This presentation was done before I had a chance to read the questions, but I wanted to put this in. So it is absolutely clear that natural infection provides substantial protection for at least several months. But the antibody response does decline after natural infection, and the strength of the antibody response does vary too, and it appears that people who have milder natural infection may have a lower antibody response compared to people with more severe natural infection. Now, we know from clinical trials that if you give a single vaccine dose to someone who was previously infected and was antibody positive, that generates a very strong boost in their neutralizing antibody titer higher than what they got from natural infection, and in fact, higher than what people who are not previously infected get after the full vaccine series. And the vaccine dose generates an antibody response that is more broadly protective against variant strains compared to the antibody response just in response to infection. So I'm calling this sort of hybrid immunity. That is, someone who was actually naturally infected and then they got vaccinated on top of that. It really looks to me like someone who has hybrid immunity probably has the best protection of anybody. And there's reassuring evidence that the vaccine side effects are not more severe in people who were previously infected. They don't have any increased risk of myocarditis or any serious problems. And so it does not appear to be a safety issue at all. And the CDC, there's a CDC study shown on the right here, or the key table published back in August from an investigation in Kentucky, where they looked at a group of people, all who had prior infection, and they looked at whether getting vaccinated influenced your risk of getting reinfected. And in fact, as shown in this table, the people who were not vaccinated had a twofold increased odds of becoming reinfected compared to people who were fully vaccinated and also previously infected. So in other words, if you were previously infected, you had a benefit by getting vaccinated that further reduced your likelihood of getting reinfected. But I do want to emphasize that natural infection does provide some protection, but I think based on all the evidence we see, you get much better protection if you get a vaccine on top of having had natural infection. Now, it could be only one dose. The recommendation is for 2 doses. So I'm only speaking for myself here, but if you just look at the data, someone who is previously infected and gets 1 dose of vaccine, their antibody levels after that first dose are higher than what most other people get after 2 doses. And so the second dose may be just a bonus in that case. Vaccine safety. We have a robust vaccine safety monitoring system in the US. One component that you've probably heard about is the Vaccine Adverse Event Reporting System, or VAERS. This is a online reporting system. Anybody can report, doctors, patients, manufacturers, and you don't have to prove that the vaccine caused it, you just have to suspect it. And so the VAERS system is a useful system because it can generate hypotheses in areas that need further investigation. However, it is a very poor system for determining if the vaccine is actually causing any adverse event. It is subject to reporting bias and incomplete data, and the fact that someone reported something after vaccination does not in any way prove that the vaccine actually caused that outcome. And so you have to be very cautious in reporting VAERS or in interpreting VAERS data. And unfortunately, anti-vaccine advocates have weaponized VAERS by misrepresenting VAERS reports and claiming that thousands of people have died from vaccines, when in fact that is not at all the case. So another sort of tool that we use to look at vaccine safety concerns is the Vaccine Safety Data Link, or VSD, because unlike VAERS, this uses electronic medical records, and it's based on actual diagnoses, not based on voluntary reports. And so it's not as subject to bias compared to the VAERS system. And the VSD currently covers over 12 million people per year. We are part of VAERS at the Marshfield Clinic Research, we are part of VSD at the Marshfield Clinic Research Institute. We have been for 20 years now. So we're actively engaged in the COVID-19 vaccine safety research. And these are the sites here in VSD. And VSD currently has well over 14 million doses of COVID-19 vaccine in our databases. So we're able to look at pretty rare safety outcomes in VSD. One thing that we're doing is called rapid cycle analysis. And essentially that's an analysis that is conducted every week to look at a large number of pre-specified events that we want to look at. Now, it doesn't mean that there's any evidence that any of these events are truly associated. It just could be they're events that were picked because other vaccines have been shown to sometimes cause it, or COVID itself might cause it, or there's just a concern about it. But essentially, for each of these outcomes, and you can see them in the table, ranging from blood clots to neurological conditions to myocarditis to heart attacks and pulmonary embolisms and strokes, etc. We compare observed number of events with the expected in a risk window after a dose of the vaccine. And with some very fancy statistical methodology, we can basically determine every week whether or not there's a signal indicating that the number of events we're observing is more than what we would expect by chance. And this is a report published in JAMA on September 3rd, and you can see at that time there was no signal on the right for any of these outcomes that we looked at. But I can tell you that since then we have gotten a signal for the myocarditis outcome shown about the fifth one down here. And the JAMA publication specifically looked at myocarditis, even though it had not signaled in the main analysis. There was a lot of concern about myocarditis because of reports to VAERS. And so this is an example where VAERS sort of provided a hypothesis, and then in VSD we could look at the hypothesis, and in fact we confirmed it, and we determined that yes, in fact, this myocarditis association is real. And on the right-hand side of this, you can see that the excess number of cases among people 12 to 39 years old after any dose was around 6 extra cases per million doses of myocarditis. Follow-up is ongoing, but data from the US and from elsewhere suggests that these myocarditis cases are generally mild. People recover spontaneously without long-term consequences. It seems to be very different than myocarditis that occurs after an actual viral infection, where it can be life-threatening and have a very high rate of ICU admission. Okay, so I just want to hit on a couple other safety issues. One is concerns about pregnancy, where the data is getting stronger every week. It was strong to begin with, but it's even better now that this vaccine is safe during pregnancy. On the left-hand side, it shows the bottom line results from another vaccine safety data link study that was published last month in JAMA, looking at the odds of a miscarriage in people vaccinated and not vaccinated. And the odds ratio was 1.02. An odds ratio of 1 means there's absolutely no risk. So this provided no evidence at all of any increased risk, and suggested that vaccination has nothing to do with miscarriage. On the right-hand side is another study just published in the New England Journal of Medicine from Scandinavia, a completely different population, where they also looked at the risk of miscarriage in a 5-week window, the risk of vaccination in a 5-week window before miscarriage, using a countrywide database. And again, they found no evidence at all of any elevated risk of miscarriage. And that's looking at all women in the population or among healthcare personnel in particular. So the ACIP does a very thorough review in making their recommendations, and they try to assess the risks and benefits. And this is just an example of the risk-benefit assessment for the Pfizer-BioNTech booster dose. And they basically model a range of possibilities with reasonable assumptions. And in this case, we don't really know the risk of myocarditis after giving a booster dose. So to be very conservative, they modeled this as perhaps the risk is double the risk of getting a second dose. Again, there's no evidence that that's true. From Israel, that's definitely not true. But even in the case if the myocarditis risk was double, If you look at the bottom here in the blue, it shows the number of hospitalizations prevented in different age groups for every million doses given. And on the right-hand side of the bottom, you see in red the number of myocarditis cases that you might expect in each age group as a result of those million vaccinations. And so you can see, just taking this very high-level view, that the benefit of the vaccine in terms of reducing hospitalizations is substantially greater than the risk from myocarditis. And then also, a very important point, though, in terms of the booster doses is that giving a million people, vaccinating a million people with the primary series is going to have far greater impact than giving a million people a booster dose. And this graphic basically shows in blue the number needed to vaccinate to prevent one hospitalization for the booster, and then in green the number needed to vaccinate to prevent one hospitalization by giving them the primary series. So the yield, the benefit is far, far greater by vaccinating more people who are currently unvaccinated compared to boosting people who already had the primary series. So just to summarize here, COVID vaccine safety is tracked in near real time by multiple systems. We've now vaccinated nearly 200 million Americans without identifying any major safety concerns. We know from doing vaccine safety research for many years that vaccine-related adverse events will occur within 8 weeks after vaccine receipt in virtually all cases. And there's basically no biological reason to expect any adverse health effects to occur months or years later. Vaccines don't cause cancer. They don't cause problems to occur way down the line. We do know that the mRNA vaccines are associated with an increased risk of rare myocarditis, especially in males under 40 and after the second dose. And the Janssen vaccine has also been associated with a rare risk of serious blood clots and Guillain-Barré syndrome. So I'm wrapping up here, and I just want to say a couple things about misinformation and vaccine hesitancy. There are many, many false claims out there on social media about these vaccines, and I hear about them regularly, and have debunked many. I debunk them as often as I can, but it's like trying to empty Lake Superior with a cup. It's like the volume is too high, and it's impossible to address all of them. And so I'm not going to read all these. You can sort of see these, but these are all things that I've heard from people. And I want to encourage people to make an informed choice, to be skeptical. And I want to, in particular, though, distinguish being skeptical from being in denial. Because I know people, and I've talked to people who are in denial of overwhelming evidence. And they say they're being skeptical, but they're not. They're just denying the evidence. Being actually skeptical is a good thing. Anyone can publish anything on the internet, and try to do your own fact checking. I put at the bottom one fact check site that's it's nonpartisan, it's not for profit. They have no skin in the game. It's a public policy center at the University of Pennsylvania. They have a section just on COVID misconceptions. It just takes a minute to do this. If you see something online that sounds a little weird, do a fact check on it. Remember that anecdotes and videos are not scientific evidence. I see many videos that are very professionally produced. They look very, to someone who's not familiar with the science, they sound very convincing. And the people talking sound very authoritative, and they seem very confident in their conclusions. But it's malarkey. Correlation is not causation. Distinguishing real experts from fake experts, avoiding confirmation bias, and basically in the bottom line, focusing on the scientific consensus from high quality studies and credible experts. So I'm going to stop at that. Thank you for your time. I'm sorry I took longer than I expected. I added a few more things, but I am more than happy to stay late and answer questions, for the next 15 or 20 minutes if anybody is still awake and interested. So thank you.
Thank you. Dr. Blagia, I went through the questions that were submitted to you ahead of time, and I believe you have addressed all of them. So I don't think I have any other questions that were in that you did not address. Natural immunity was one of those. Easing the concern of parents with young children who are afraid of the side effects such as myocarditis. You have that information available. I think the low health literacy, I believe that how we help those individuals, I believe the Department of Health Services has provided information at as low a literacy as they can. And I also know there's the other organizations, probably the Wisconsin Literacy Organization, I believe also has materials that are written at a lower level. The goal was not to, we want to make sure everybody understood what's being done. So I'm seeing a lot of thank yous and not so much. Here's a question. What do you feel about continued mask use, children in schools to decrease the spread? Any thoughts or?
Yeah, no one likes it, but it is a safe thing to do, and we're still at the point where we're just right now. It looks like maybe we're kind of emerging from the most recent wave, and now, fortunately, it looks like things are going down. But for the time being, given that children are unvaccinated for the most part in school, the mask use is an important mitigation strategy. Ultimately, vaccines are going to be the way out of this. But for right now, I think the mask use is very important.
Mm-hmm. I know WNA for sure wants, would like to see that we continue to use in those, now that winter is coming, we're going to be indoors more and, trying to get out to the shopping centers, that it's going to be important. I think that we, at least for us to protect ourselves from others, would be the masking. Do you, so with the kids, they've been doing the studies, and so the ages 5 to 11, just the side effects were not any different than for the other age of children in terms of side effects or?
Yes, that's true. So just to bring everybody up to speed, the FDA reviewed the data from Pfizer on their clinical trial of children 5 to 11. They basically had a close to 5,000 children. Two out of 3 children actually got the vaccine, and it was a lower dose at 10 micrograms instead of 30, so it's a reduced dose of the vaccine. It's a separate formulation compared to the adult version of the vaccine, and they found that it was 91% effective for preventing COVID-19 illness, and it generated a very strong antibody response. And the safety profile was very similar. They have data, of course, already on people 12 to 15 years old. So comparing the younger children with the older, it looks to be very similar. Okay. And there were no episodes of myocarditis at all. And including in children, there was those that were previously infected, were seropositive. They did not have any increased risk of having any side effects or severe reactions. So everything looked very good there, and so and the FDA advisory panel signed off on that and basically said they recommended the FDA authorize it. The FDA did, and now the ACIP will be meeting next week on Tuesday to discuss what they want to recommend, and they go through a sort of an additional process, and they look at the feasibility, the risk and benefit, obviously, and the scientific evidence for benefit and any potential risk, but they also look at things like implementation feasibility and the overall impact and on the epidemic, and to the extent that this is an important public health problem. So they're going to go through sort of a much broader review on Tuesday that will end up in a vote. And then if accepted by the CDC director, which it almost always is, that will then become the national policy and recommendation.
Thank you. That was very, very helpful. And again, because of the kids, it's important that we get that information out there and accurately. So this is a question about, vaccinated individuals hanging out with vaccinated individuals. We know the holidays are coming. Any thoughts on masking if you're hanging out with the family?
Yeah. Again, I want to preface this. I'm just giving you my opinion here. I'm not representing any official recommendation. So I think it depends on your risk tolerance, ultimately, really, because you get into gray areas where there's no real clear answer. If you have a very low risk tolerance, then you might want to wear a mask. On the other hand, if everybody in this space is fully vaccinated or even boosted, if they're eligible for a booster, we know that each of those individuals has a substantially reduced risk of becoming infected and shedding, but it's not zero, but it is substantially reduced compared to unvaccinated population. So, if one person happens to be, despite the lower risk they're infected, the other people are vaccinated, their chance of getting infection is low. So when you start adding up these probabilities, the likelihood of you getting exposed in an indoor group of people where everybody is fully vaccinated and nobody is sick is pretty low. But again, it depends on your risk tolerance. And so if pretty low isn't good enough for you, you want it to be as close to zero as you can make it, then you might want to wear a mask. So I think I'm going to dodge the question a little bit.
That's okay. And that's fine. I thought that was a great answer. So where do you see the future of more the use of mRNA in other medical treatments, or has there been discussion about that?
I think it's going to usher in sort of a new golden age of vaccine development that has been kind of stuck for a long time. And this crisis has given a jumpstart to this new technology, and I think we're going to see spin-off benefits of this in other areas like influenza and RSV, which is an important respiratory virus in young children especially, and older adults. And there are already vaccines in development for other things as well. So I'm very excited about the potential for our mRNA, and the nice thing about mRNA is these vaccines can be scaled up and manufactured quickly by companies that have the expertise in the facilities. But a big issue that I didn't really talk about is that most of the world is still unvaccinated, and most of the world doesn't have access to this mRNA technology. And even though conceptually, the way I showed it, it's not that complicated conceptually, the manufacturing is very complex. And it is very, very different than any other traditional sort of vaccine manufacturing. So plants in India, for example, that know how to manufacture other vaccines, that doesn't help them much for manufacturing these vaccines. And so I think there's going to be a need to sort of export this technology and this capability, because right now, with only 2 companies really doing this, they can't meet the global demand. And so we have to scale it up and make this technology available throughout the world.
Somebody asked about why are Wisconsin counties still so high, that even Dane County, we saw a spike in the Delta variant.
Yeah, I don't know. There's a lag in reporting. And so, what we're seeing now may be reflecting transmissions that occurred several weeks ago. And we know Delta is far more transmissible than the original strain and even the previous variant strains like the Alpha variant. And so if you take the fact that the Delta is extremely transmissible to the point that, you're either going to get vaccinated or you're going to get Delta, it's not too surprising, but we are seeing this trend coming down in Wisconsin and certainly nationwide. I don't pretend to know exactly what's driving that, but I hope that continues, and I hope that we start to see reductions, throughout the state.
I think we all hope for that as well. We all know that probably there's not a healthcare provider that is not tired and has been working so long on this. And someone said, is there a new mutated virus? I heard about, is it the Delta Plus?
Yeah, there's Delta Plus. Sometimes the media sort of latches onto things maybe a little prematurely, and Delta Plus might be in that category. It accounts for a very small percent. Apparently there's some very inconclusive evidence that it might be somewhat more transmissible, but it's not at all clear that it's going to take over or become more common. At this point, we just don't know. If you talk to the virologists, I think what I'm hearing is that the next sort of stage of this is going to be that so many people in the population are immune either from being infected, they have immunity from infection, or from vaccination, or both. That immunity is going to sort of drive further evolution of the virus, kind of like it does with the flu virus. And so the further changes may not be sort of changes that increase the transmission risk, but changes that sort of evade immunity. And we just have to wait and see. Nobody really knows what's going to happen on that.
Okay. I think maybe we will stop at this point. There are still a few more questions, but I think if we just advise people to just go to the DHS website, they'll find the answers to many of these. And like I said, we would, if you as a WNA member join our Wisconsin Nurses Connect, we've set up a specific site or a topic that allows you to join the conversation, and we'll put some more of those answers in the chat box. But I would very much like to thank Dr. Belangia. I don't have it yet, but and for your time and your expertise. And I just asked Bree to put up one more slide, the very last one, just to, here we go. All right. Just again, so you are going to get an email either this evening or tomorrow that will give you the link to the evaluation, and from that you will get your continuing education. For those of you, we hope that we found this, I think we all found, I found this very informative and supportive of our practice. And like I said, we'll continue the conversation. For those of you that are not WNA members, it would be shameful if I didn't bring it up. So if you are not a member of WNA,
he crossed it and he pushed him back. He was across. He crossed.
And that our membership dues are $15 so a month. So please consider joining ANA and WNA. And again, thank you all. We really, really appreciate your time. And I think you addressed so many of my questions. And I think you helped a lot of others. And there are your presentation slides already posted on the website. Just incredible information, and you're obviously very good at what you do. We really much thank you for being part of Wisconsin's trying to get us out of this world called COVID.
Yeah, well, thank you for having me. It was a real privilege, and I really enjoyed it.
Okay, thank you all. Okay. Have a wonderful night, and I guess we say go Packers. I don't know. Two of the players are out with COVID.
Go Packers, go Badgers.
Yeah, and Badgers. Thank you. Okay. Good night.
Bye.